The Fish and Invertebrate Lab at SERC has been studying the movement of river herring in the Chesapeake Bay since 2012. River herring spend most of their lives in the ocean, but swim into streams to spawn. Researchers believe that dams, which block herring from reaching their upstream spawning habitats, were a major contributor to the population decline.
The goal of our project is to understand how big the populations are and what places all around the Chesapeake Bay watershed they're using to spawn.
This is a good project. We like to bring in volunteers. They help us out in the field collecting data, and they also help us back in the lab analyzing these samples. One of the aspects of the river herring project is habitat use. So we'll go around to different tributaries of the Chesapeake Bay and sample small creeks in those areas, and we'll look for presence absence. And we do that 3 different ways. We take an ichthyoplankton sample, which is just collecting fish, eggs, and larvae with a plankton net. And we also do a visual observation, can we visually see them here or not? And then we take a water sample, which we use to process eDNA, so environmental DNA.
The eggs and larvae are great for telling us if they're fish spawning, but one of the challenges is that we can't tell if those fish are one of the 2 river herring species, alewife or blueback, or they could also be hickory shad. They all have eggs that look pretty similar. So it gives us a good idea if the habitat's being used for spawning, but doesn't tell us exactly which species is there.
In 2017, our researchers went to the Elk, Bohemia, Sassafras, Chester, Pocomoke, Patapsco, and Potomac rivers. Citizen scientists and researchers helped collect ichthyoplankton samples at all of these sites. Of those rivers that we sampled, we found ichthyoplankton evidence at 5 rivers. This is a map of the Chesapeake Bay. The blue dots indicate sites that our researchers and citizen scientists sampled and found possible herring eggs and/or larvae. The red dots indicate sites where we didn't find herring eggs or larvae in the sample.
The environmental DNA sample seems to be the most sensitive method of detecting herring presence absence so far. If herring are in the water, they are shedding some sort of DNA. So we are able to tell more definitively whether herring are present or absent in this system.
Scientists in the past have only used eDNA to determine if a species is present or absent. This year, however, with the help of the University of Maryland, we've been able to determine which species of river herring, either blueback or alewife, is present at a particular site. By understanding which habitats the herring are most likely to use, regulators can better prioritize habitat restoration to best conserve and restore river herring populations.
I'd like to extend a thank you to our volunteers. This project would never be possible if it wasn't for you. It's lots of hours in the field, lots of hours in the lab, looking at our microscope, not always doing fun things, but if it wasn't for you, we would have never been able to accomplish it.